Thank you, Dr. Janet, for her wonderful speech. And we will start with the biorefinery section through one of our Dr. Mariella Rivas from the Biotechnology Lab Algal from the University of Chile, the Autofagasta University in Chile. So welcome to Dr. Mariella Rivas. Thank you very much.
Hola, mucho gusto, buenas tardes. A los organizadores invitados y a los organizadores también que este evento. Bueno, yo soy de la Universidad de Autofagasta, no de Autofagasta, y de CICITEM, así que tengo doble filiación. Vamos a cambiar radicalmente de tema y voy a empezar con el tema de la biorefinería de microalgas. Para ubicarlos un poquito, nuestra universidad está en el norte de Chile, por lo tanto...
I'm sorry. This is an area that is very full of desert. We have several advantages. We have a lot of intensity, light intensity. The climate is template, and it varies very little during the whole world, even through the stations. And we have the advantage and disadvantage that we have a lot of companies that produce a lot of CO2. Here, worldwide, we have one of the regions that has the most intensity of light compared to Africa. This is our peninsula, and if you see, this is a desert. For this reason, the microalgae here, they don't fight with lands available for other kind of agriculture, so the use of land would create a feeding competition. I'm going to talk a little bit about the microalgae. Mostly they're small microorganisms that are photosynthetic, whose habitats are very diverse. They have agricolas, marine, rivers, lakes. They are responsible of 50% of the primary production, and they are the base for the production of food. Here we can see different types of microalgae that we have been able to, like 7 years ago, we had a new rediscovery because their biomass can be used with bioenergy, like biodiesel or biogas or biodiesel, but it can also available or good for the development of human consumption, animal consumption, to have therapeutic products, or for the treatment of water through bioremediation. They have the ability to adapt to different types of environments, and they have a large variety of lipid grease. They also synthesize a whole bunch of different elements like vitamins, They have a high rate of growing, so in a very little time we can have a lot of biomass. We're able to use secondary waters, and they consume CO2 for their growth. Here we have some of them. We have the Biococcus, which is one of the responsibles one that um, a review was published that this algae has 80% of the oil, which is true. And it also has the advantage that it's can be done in a massive way, and it can represent the cost benefit, economic cost benefit, a whole lot bigger versus cultivating other type of mass. All this depends if the applications that you want to give to it. For example, if you want to be able to get biomass for gas or for economic use, one should use reactives, uh, cultural systems, which are photoreactors, which are very economic. Now, if we want to be able to get products and subproducts with more reactive issues, then yeah, you have to have a little more care with how to produce it. And then these kind of systems that is closed, they're very inexpensive. They're plastic bags, but there's some that are done by acrylic. There's a big development of technology to be able to produce algae worldwide. When I started working with microalgae in the biodiesel production, and now we're into the bioproduction of reactive products. Now we've more or less since 2010 we've been working on the production of biodiesel or bio um, oils for industrial application, where we were able to put these type of growing and with a thermoelectric plant, and one of these chimneys, the gases of CO2, and they were injected directly into the crops of the microalgae crops, and they had an amazing productivity constant of about 1 gram per liter day. So to be able to put a bioalgae, there is a very big necessity to control the different processes, uh, and we also need a research that has to be integrated with not only with microbiologists but also with engineers and with automated systems. It is possible that this can be done by hand, but you need to have an injection of CO2 that has to be automized. Now that was like kind of the introduction, and now what we have done in these last years. The first I want to talk about is that in Chile, energetically, it's a dependent country because we import all our energy. So then the country decided they have to 20% of their electrical had to be 20% of produced by themselves, um, and we are very close to it right now through solar energy and bioalgae energy. But still, we're 2011. We can see it represents a 1.5% more or less of all the renewable energy, non-conventional. And talking about a little bit of the biogas, it's an energy that is produced naturally in all the environments. It's produced by bacteria and archaeas through a process that's called an aerobic process. It has 4 phases. If you look them in a book, you can find them. It's 4 phases, where the last phase is the metallogenic phase, which transforms and produces all of the organic residual and methane and CO2 with components, nitrogen and sulfur components. For this reason, the biogas, if we have maybe water that is stuck and you want to produce it naturally, the cows also produce through their residue, and then we produce it industrially. This is the plant of Metrogas in Santiago, where all the home solids are received, and it's been able to work with 30,000 homes. This is right now. We have a problem we have at this moment. In this region, has sanitary problems, has a very big garbage, and they burn the garbage, which affects the environment, and it doesn't have a structured garbage system. So all the residues just stay there, and so there's a production of biogas of these residual would be a good solution. We're trying to be able to go through a process, taking advantage of all this garbage and now all these microalgae to be able to get this. And just since the garbage by itself does a very bad level of biogas, and then with the algae it would help to make it a better quality. This is in general. I'm going to go a little more detailed. I'm going to talk these 3 different projects that we've been developing during these years. So under this concept, we have residual waters with Biorefinery, where we have microalgae, and then we get biogas. There's several papers you can find in the different literature where you can use different types of microalgae to be able to produce this gas. And here you can see more or less that it's in the order between the 300 to the 592 ml. So then if we take into consideration 1 kilogram of microalgae can produce more or less 485 kilograms of methane, which would mean 27 megajoules of energy, or 8.6 megajoules of electric energy. And this is because of the chemical composition of the microalgae, because of the lipids and the proteins that are at the mumus. These are some of the results that we have been able to get during these years. We've been able to get biogas made from these microalgae, which is possible, and it all depends on how it is cultivated. For example, this one, it produces methane up to 400 ml, but the microalgae, it's very difficult, and it takes 30 days, so it's very hard to get the biomass from them. And it grows in sweet water, which is very little in the desert. So therefore, we're talking about microalgae, marine microalgae, which is also being injected with the water, seawater, and then we're getting similar levels of methane. At some point, we thought that the residual salt of these cultivations, we thought would inhibit the growth of the microalgae, but we have discovered that there is no interference. And now, this is specifically what I've been working on myself, which is by using microalgae, since they're vegetables, they have a cellular wall, which is a physical barrier so because of this, the production of the biogas goes down. So we have new alternatives. One was try to obtain some kind of economic method to be able to break this cellular wall, which is composed of cellulose, and some of them have what is called the algae, which is very resistant, the cellulose, resistant cellulose. It's a polysaccharide that is made of glucose, and it has a life of 8.5 million years, so it's very resistant. So to be able to produce biogas, we have realized that we have to have a pre-treatment to the bioalgae, and then when we use these different kinds of treatments, which will put the production of methane higher during the time. So what we did was work with that in the world there exists, in the different ecosystems, bacteria that can help degrade these microalgae. So we were thinking that they were in the mollusks or in the herbivores, and what these animals do, they feed themselves from phytoplankton, and they usually have a high percentage of microalgae. So we started looking there to see if they had some kind of bacteria that could have a special activity that would help us degrade the cellulose of these walls. And what we were able to find, we found more or less 11, which are the ones that we characterized with some methods. Usually these methods, to be able to detect this was a positive control. We were able to evaluate the filter and if it degraded, and then this was put, we decided a wholesale where the bacteria with cellulosa were mixed with the microalgae, and then we had a recirculation until the microalgae had been more or less degraded. And we did the microcycle and at the 48 hours we were able to do a fluor-white calculations, and we were able to see some kind of degradation. And this was used. This is obviously we had a larger process. This is very simpled out. And then we had this treatment, and then the production of methane went up in about 20%. Therefore, we realized that that was very important. So for this reason, the other alternative that we used was to do this pre-treatment and to involve another step of the treatment. So we tried using another microalgae that has been really used, that is with the Carti, as part of the study. And so since it's so well known, they were able to characterize, and it mutated. And we have this one. It has a deficient cell wall, and we cultivated it outdoor, and it grew very fast. In 7 days we had the biomass. We did the anaerobic system, and we realized that by using a mutant that we have right here, that's represented in the green, with respect to the other algae, there's a higher production of a biogas. Here we can see the methane. This represents the methane, and it's 40%. And the cepa that we use with the microalgae with a weak when it comes to 50% when it has a weak cell wall, whereas with a strong cell wall it only comes up to 40%. So then we've been able to go out, get the efficiency going up by using different kinds of microalgae. And here we can see how we compare it. Here is with the weak one, and here we've compared with the other two. There is a difference, the same amount, it gets more or less the same amount of methane. Finally, to be able to do a bioprospectation or biorefinery, we are looking also for native algae of the area, which are very strong, extremely tolerant, and that tolerate high temperatures and high lumosity. And therefore we have gone to different parts. You can see from the pictures down here that we used these rugs, which is under the salt, and we have been able to isolate microalgae from there. Right now we have 40 different cepas isolated in different parts, and we have totally characterized, and we have a bank of cepas of 200 more that we have to characterize. It's a challenge to be able to isolate new cepas, but then we've been able to characterize them and be able to cultivate them in standard laboratory conditions. We found some bacteria and some algae that have a high context of pigment, and also higher productivity and high growth rate. And additionally, it has a high contents of carotenoids and also the cicosanina. The kilo of cicosanina costs around $24,000. So then if we want to work in the biorefinery, we have to combine the mixture of which would help us produce energy at a low cost. These are the same samples. We have this one right here that we get to 60% of the pigment. Any other questions? You can ask me afterwards. Okay, thank you very much.